Okay, I will get started. So thank you for joining me today for this BDRC Intro to BDRC webinar. Like I mentioned, if you haven't already created an account, I would recommend you do so. It's free. All you need to do is go to bdresearchcloud.com. If you already use FlowJo portal to log into FlowJo, it would be the exact same email and password credentials. So if you have those, just put those in and press sign in. If you do not use FlowJo portal, not a problem. Just hit this create account button and enter your email address and follow the instructions there to create a new account for BDRC. On the agenda today, we'll first do an overview of BD Research Cloud. What is it? And then we'll go over the internal structure. So what are groups and projects and what do they represent within BDRC? And I'll help explain how to set up your organization. Then we'll go into the demo and use BDRC, and we'll go through how to create an account and how to invite users to this account, how to create groups and projects. And then we'll go through all the resources, how to upload and save a cytometer, create populations, create reagents. And we'll use all three of these resources during the panel creation. Another thing you can do within BDRC is to create a FACSDiva experiment and also upload data and connect to FlowJo for analysis. If there are particular items that you wish to spend more time on, just drop them into the chat or the Q&A. I'll be monitoring those during the presentation. But if I don't hear back, I'll just kind of do a general overview of BD Research Cloud as a whole. Let's start off with what is BD Research Cloud. It is an online ecosystem supporting flow cytometry studies. So what can you do within BDRC? You can upload, save, and share cytometer configuration. So if you run a core lab, you can upload all your cytometers to BD Research Cloud to more easily share with your users so they can use that information to create panels to use these cytometers to acquire data. These can be BD cytometers, but they don't have to be. You can upload any cytometer with any configuration to BD Research Cloud. Another thing you can do is create panels using reagents from either the BD catalog or custom reagents. Again, these don't have to be BD reagents. You can upload other reagents from different companies. You can manage your reagent or antibody inventory and track titration information. And you can also view the spectra of the fluorochromes within the various spectrum viewers. You can also use workflows to keep track of who did what and when within your lab. These can be panel creation workflows, but they can also be experimental workflows. These are completely customizable. You can keep track of if anyone performed a cytometer clean or QC that morning, or also keep track of more protocol-like experiments of who stained the cells, who thawed the cells. You can upload data of the cell counts, things of that nature. In BDRC, you can also create FACSDiva software experiments. So when you're ready to acquire your samples, you can just hop onto BDRC and get a leg up to label your samples ahead of time away from the cytometer. This is particularly useful if the cytometer is in a core lab that's in another building. You can just log into BDRC from anywhere and start your experiment creation directly in BDRC. And lastly, you can upload your data. So once you're done acquisition on the cytometer, you can upload your data directly to BD Research Cloud and connect to FlowJo so that you can perform analysis. Your files are available to you from anywhere. And then by connecting to FlowJo, it manages your files for you, depending on which workstation you're looking to analyze. So a quick note about structure in BDRC. When you first sign up to your account, you are given an individual organization. So that is for you and it's for single use. You cannot invite other people to your individual organization. But you may also create a separate organization that's for your lab. So that would be a collaborative organization. So this is the individual organization that's created for you automatically when you create an account. If you choose to create a separate account for your lab, then you can add groups to that organization. And in BDRC, a group represents users working together on one or more projects. Putting users together in a group means that they'll have access to all the data from that group. So if you're just creating an org for your lab and you want all users to have access to all the data you upload to BDRC, then I would suggest just create a single group, put everyone in it, and then you can organize your data for under different projects of that single group. Projects are found within a group and contain all relevant files, workflows, and data. Users within a group will see all the projects and data from that group. And when you invite users to your organization, you must assign them to a group. So that will dictate what data they have access to. A user can belong to one or more groups, and there are no limits on the number of groups within the organization. Groups and projects help you organize your data and determines who has access to it. Again, if there are any questions, please drop them into the chat or the Q&A. So these are two different ways you can create an organization using BDRC. If you have a large institution and you want everyone within that single org, then you would use groups to separate out different labs or departments, and you would put users within those different departments. And all the users within those labs or departments will have access to all the data within those. So that's the case if you want to include a very large amount of people within BDRC. Alternatively, if you just want to keep it smaller and just have your lab within as the organization, then you can have groups within that lab and you can separate people based on those groups. I would recommend having at least one group where everyone is in it, so you can have common files and folders that's accessible to anyone. And then you can choose to make additional groups. For example, if you have a collaborator that you want to share data with more easily, you could create a group and the collaborator will only have access to the data that's relevant to them, and you can separate out your other data and it won't be visible to that collaborator if you put them in a separate group. So that's just an example of how data is organized within BDRC. If you choose to add a cytometer to your lab, it is available to everyone within your lab, independent of the group. So the cytometer lives outside of the group. Everyone will have access to it. And you can also choose to make it publicly available. So again, if you have a core lab and you want to share your cytometer details to other members of your institution, but you don't necessarily want to invite them all to your BDRC organization, they can create their own BDRC for their own labs, but connect to your cytometer. All right, we're jumping over to the demo. All right, so that was the brief overview of what is BDRC and how to stay organized within BDRC. I'll now make this bigger. And let's log in. Again, it's a bdresearchcloud.com. If you have FlowJo portal credentials, you just pop those in. If you do not, you hit this create account button. When you create an account, it asks you for your email. Just put your email in there, press sign up, and follow the instructions. But if you do have portal credentials, you can just pop those in and sign in. So the first time you sign into BDRC, you are logged into your individual organization as listed up here. And like I mentioned, this is for you to use. You cannot invite other people or share from it. And so if you go to people, users here, it says your individual organization is just for you. To invite users to collaborate, click your name in the upper right and select create an organization. So now we'll create an additional organization for your lab or for the group of people you want to work together with. So I'll click my name here and hit create an organization. Here we see the different license types for BD Research Cloud. The individual is the one that's automatically created for you. And then we have the essentials license that can allow up to 10 people to collaborate together for free. So this is a free organization. Along with this essentials license, you also get additional storage per user and a greater amount of workflow limits. Here we can see which features are available in the different license types. So in the essentials one, you can create workflow collaborations and user-based permissions, in addition to the individual features that include experiment workflow tracking, BD FACSDiva experiment creation, data and metadata management, and the reagent selector panel creation process. There are 2 additional license types, the professional and the premium, and they include additional features. The price for these depends on the number of users and seats, similar to the FlowJo model. So depending on the number of users you choose to add to these license types will determine the cost. So just click for quote to find out more specific pricing for these. But in the professional license type, you can have automatically recurring workflows that's not available in the essentials license. And then the premium license type, you can have the reagent inventory management feature, antibody titer management, and also the ability to share files with people that are outside of your organization. And then there's also additional storage that comes with these license types. But if you're unsure and just want to try the free license, please feel free and click create free organization. So you click this button and then you give your organization a name. So if it's for your lab, you would put your lab name here, or if you have a core lab, you'd put your core lab name here, and then fill out the additional information and press submit. So once you've entered this information, a new organization will be created here. So in addition to your individual organization, you can have a drop down, and your additional one will be listed here. I have a particularly large amount of labs here, but I will just jump over to this one, my lab. So once we are in this collaborative organization, what I recommend users to do is go ahead and create a group to invite users to. So if I hit people, groups, and projects, I'll see the groups that I've already created. If you don't have any groups, you can hit create group, and then just name the group. So this could be the common group, or just name it whatever you want. Again, this will dictate the people within this group will have access to all the data from this group. So if you're just creating one for your lab, I would say just name it as such and invite everybody to that group, and you can create additional groups here if you want to isolate certain data to be restricted to just some people. Once you've created a group, you can click on that group and you'll see the projects that live within it. Again, there are no limits to the number of projects. So if you have that one single group for your lab, you can then name the different projects based on what's being studied in your lab, and you can save data under these different projects. Any questions so far? Feel free to just tap them in the chat. So once you've created groups and projects, now is when I would recommend you invite users to your different groups. So under users, you can hit invite. And you would just simply put the email addresses of the people you choose to invite to your group. Then you would select which group you want to invite them to. You can select as many groups as you want from this list. So you can add 1, 2, 3, doesn't matter. So again, a user can belong to one or more group. This will dictate what data they have access to. And then along with this invitation, you can determine what permissions you want this particular user to have. So do you want them to be able to manage organization level permissions? So do you want them to be able to create new groups and projects? Do you want them to be able to manage cytometer inventory? So change cytometer configuration information, manage custom reagents and inventory, view workflows outside of their own group, view and edit files outside of their own group, or delete files within the organization. So depending on who you're inviting and what role you want them to have within BDRC, you would just turn these on or off. And then after adding the email address here, you would send invite. And if you want to invite multiple users at once with the same permissions, you can. You just add them separately. So here to enter multiple addresses, you can just add them one at a time or add them all at once. All right, so now that we've created groups and projects and invited users, oh, here we go. We have a question. Does the user need to have their own account before, or is this an email to invite them? Good question. You can invite them even if they don't have an account yet. So what will happen is when you invite them, you put their email address, they'll receive an email from BDRC saying, so-and-so wants to include you in their org, click here, and there's a link that will bring them to BD Research Cloud and they can sign up at that point. Good question. All right, so then once we've created groups and projects and you have a list of users, you can add more resources in preparation to create a panel. So if we go to the cytometers section, this is where you can either add your own cytometer if you have one in your lab or if you're running a core lab, or you can also connect to a core lab cytometer. And in both cases, you would hit this create button. So by pushing the button, you can see there are 3 different ways to add a cytometer to your organization. The first way is to import a BD FACSDiva file. If you do have a BD cytometer that runs FACSDiva, we recommend this route because then it'll import all your parameters with the correct spelling and there won't be any typos or mistakes. So we truly recommend this option if that is the cytometer you have. So in Diva, you just go to cytometer configuration, I believe, and export that file. And this is what you would import here. You would have to just give your cytometer a nickname, an optional description. You upload your file here that contains the configuration information, and then you also assign it a lab location. So you would just click here, add a new lab location, put the lab location details. It could be a room or building number, additional description, if you choose to add that. And then here, this checkbox, publicly discoverable, this determines if you are sharing this cytometer configuration with anyone in BDRC. So if you're running a core lab or you expect outside users to want to use your cytometer, you would check this box and it'll make it available to users to discover your cytometer, to create panels with it. They won't have access to the configuration, to editing the configuration. So you don't have to worry about access in that way. They'll just be able to view the configuration information. They won't be able to modify it. So that's what this means. And you would just press add cytometer, and that would be the first way of saving a cytometer. The second way, if you don't have a BD instrument, or if it's not running Diva, you can create a custom cytometer. And in this case, you can either manually create it. So you add a lasers, you say, you know, this is a blue laser, it's a 48 wavelength with 100 nanometers. And then you would just put in additional detectors. So yeah, this is a 530-30, for example. You can name the parameter, add additional detectors, and add additional lasers. You can do that manually. Or if you want to use another cytometer as a base and just make small modifications, you can do that as well. So let's say if I start over here, I remove cytometer details. And if I want to use a different cytometer as a base, I can do so. So we have all the cytometers I have connected to already I can use as a base, or any of the system cytometers, which are the BD lines of cytometers. So you can click on one, select a configuration if there's more than one, and it'll pre-fill this information, and then you can just manually make any changes if needed. You would give this cytometer information a go, rename it if need be. The model here is quite important, that if I just delete here, make sure that the model you select is available here from the drop-down if it's a BD instrument. If it's not a BD instrument, not a problem. But if you want to be able to create Diva experiments, we need this model to be compatible. And so make sure you just select it from the drop-down if it's a BD instrument. Again, you would select a lab location and then choose if this cytometer is publicly discoverable or not, for others to view or not. Whoops. And then once you're done, you would click add cytometer and be finished. And then lastly, if you're looking to connect to a publicly available cytometer, you'd go to this tab. So this is where you would look up your core labs. So if you click on here, you can see the different core labs that have already uploaded their cytometers and have chosen to make them publicly available. So if you do the same with yours, that you check that check mark, it will show up in this list. So if for fun, I'm going to pick the Fred Hutch I will see, oh, here's their cytometer. I can click on it. I can decide if I want to, if this is the one I want to connect to or not. If so, I would just press link cytometer. And by pressing link, then it adds the cytometer to the list of my cytometers here in the main page. So those were the 3 ways to create a cytometer or to link to one. If I chose the wrong one, or if I want to unlink it, I just click on it again. Whoops. Actually, if I want to unlink, I click on it here and then unlink from here. So now it'll remove it from the list of available cytometers to me. So in this tab, my cytometers are the different cytometers that I've either created or linked to. The ones that I don't own myself have this little green tab, public, whereas the other ones that don't have the green tag, those are the ones that I've created and uploaded myself that I'm managing. So those are the cytometers that show up on my cytometers. If they're compatible for panel creation, all of these will show up in the drop-down list during panel creation. In design are cytometers that I've started to create, but maybe I got interrupted. I didn't have time to finish updating all the configuration information, and I'm not ready to publish it yet. So the last step of a cytometer when you hit Save Changes is to publish it. Once you hit publish, then it becomes available for other users. But if you don't want to publish it just yet because you're still uploading the information is not complete yet, that's where the cytometer will live in design. So it's right. Remind me, you haven't published it. Do you want to continue without publishing? Yes, because I'm not done entering the information. The last tab here are just system cytometers. So these are pre-configured BD cytometers that you can either use as a base or use as is to generate panels with. Any questions about cytometers? If we move on, we'll go briefly over these additional resources. If you want more information, I'm happy to spend more time. So in the resource populations, this is an area in BDRC where you can define populations of cells with sets of markers. So if you're always identifying cells using the same set of markers during panel creation, then you can predefine them once in this section and then use those during panel creation. So BDRC comes with literature-defined populations already preloaded. So anyone who's created an account in BDRC will see these populations. So if I click on this first one, B cells, CD19, it shows me that for these cells, they're identified by being CD3 negative and CD19 positive. Similarly, for monocytes, they're CD3 negative, CD19 negative, CD14 positive. So if I choose to create a panel using any of these populations, these markers will get pulled in with the antigen density information assigned to them, so that they're negative for CD3, negative for CD19, and positive for CD14, assuming you want to stain for all these markers with your panel. I've created a couple of custom populations, and with those populations, you can, I'm not sure which one I was select, you can assign different antigen densities. So these can be low, medium, or high for CD3, low, medium, or high for CD4, negative, positive. So you can add all this information ahead of time. This is particularly useful if you have new lab members in your lab that aren't sure about all this information. You can have your senior lab staff input this information, so it makes panel creation a little bit easier. All right, we're going to move on from populations. But just, yeah, if you wanted to create a new population, you would just hit this create button, enter your population name, target species, and then add your markers one at a time. You know, for example, CD3 would come up here. You would assign it which expression level you want to give it. So let's say it is high for CD3. Then it adds it one at a time, and you would just keep adding to this list until you are happy with the population you're looking to create. Okay, so moving on. We're going to go to the reagent section. So within BDRC, you have access to the entire BD catalog if you want to look up quickly if a particular reagent is available with a given fluorochrome for you. You can also create your own custom reagent. So when you are creating a panel in BDRC, the list of fluorochrome gets populated in two ways. So one is from the BD catalog and the other is from custom reagents. So this is where you would add reagents that either are not made by BD, so you can add competitor fluors here, or if you have any custom reagents such as like tetramers that you've made in-house, or fluorescent protein if you have a transgenic mouse, anything like that, you would just create it here under custom reagent, and it will then show up during panel creation. So these are the two tabs that populate your fluorochrome choices during panel creation. We also have a spectrum viewer that you can choose to select any of the cytometers that you've linked to. So either the ones you've created to or that you've linked through the publicly available option, you can select those. And then you'll be able to look at specific fluorochromes to see if they're a good choice for the cytometer you're looking to use. So here, you just click on the laser line. Full line shows that you've selected the primary laser for this particular reagent. And then a dashed line shows spillover from cross laser excitation. So Percy PSI 5.5 gets a little bit excited by this laser, by this violet laser line. So it will show up on these filters for the selected cytometer. So that's a quick way to look at the cytometer configuration and fluorochrome compatibility. During panel creation, this gets expanded as well. And then lastly, this tab, My Inventory, is available for premium license types. So here we have premium and we have reagent inventory management and antibody titer management. So these are premium features. Again, we're happy to provide you with a free premium license if you want to try it out first before committing to buying a license to see if it's useful for you and your lab mates. So in your version, if you've only created an essentials lab, you might not have access to this tab. But if you want access, just contact me and I'm happy to upgrade you to try it out. In this tab, what you have access to is you can add specifics about your reagents. So quantities that you have in your lab, how many vials of this reagent do you have, the lot, the expiration, the remaining volume, but you can also add titration information. So once you've performed titration on this reagent, you can enter this this amount here so it's available and readily accessible for lab mates to view. At this point, I'll mention the linking feature. So you may have noticed this icon situated in various places through the web, through BDRC. And this is just an easy way to link items together. So here, these are gray, which means that they're not occupied, so it's blank. But once the color changes to blue, it shows that there's an item within that that's linked. So in this case, CD11 BUV 395 has a linked item to it. And if I click on it, it shows that, oh, it's a PDF. So if I click on it, it'll bring me to that PDF and I can view it. And here I've uploaded a PDF of the titration information for this particular reagent. So that's one way to stay organized within your lab and easily have access to relevant information of your reagents. So if I go back here to resources reagents in my inventory, so that's how I got to that PDF. If any reagents have expired, it lets you know. And then you can also add titration information, as I mentioned. You can add additional information. So this is 2 microliters within a total staining of 400, which is a 1 in 200 dilution. It was stained for 30 minutes at 23 degrees. It was intracellular. You can add additional notes if there was a specific protein or lack of protein that was needed. And you can also choose to create multiple titers for the same reagent for different conditions or different cell types. As needed, so you're not limited to one. If you do have more than one, you select which one is shown on the main page. So this display on card selects which one is shown on this main card here. Ways to add reagents to your inventory is the simple add to inventory. So here it shows this one's already in inventory, or you can add to inventory. Same with custom reagents. If you add them to your inventory, it says how many vials you actually have, and you can add titration information. You can also export and import your inventory for easy bulk additions. Same with a custom reagent. So you can create a custom reagent either one at a time here, or you can create multiple with this export import. So you just export the template, add more information, and then re-import it, and it'll create new reagents here that'll be available during panel creation. If there aren't any more questions, I will jump into panel creation. So if I go to the panel section, BDRC comes preloaded with OMIPs. So these are published, pre-validated panels that other labs have optimized. So they, like I mentioned, they have a publication. If I click on this OMIP 80, it lists what reagents were used in this panel, has a link to the citation. So this OMIP 80 is a 29 color flow cytometry panel for evaluation of NK and T cells. So if I go back here, I can click on the cytometer to see which cytometer it was optimized with for the paper. So in this case, it's a 5 laser instrument. And this was the configuration. So I can either choose to use this panel as is, or I can choose to use this panel as base and modify it. So in addition to OMIPs, you can find BD panels as well that we've pre-optimized for you. And in this page, you can also filter by antigen. So if you want to see all the panels that include CD3, maybe 56 for NKs, right? As I add these, the list gets filtered and will only show me panels that contain those markers. So if I, trying to think what else I could filter by, CD14 as a monocyte. So you can see that you can narrow down of which panels might be optimized for your needs. So that's one way to filter panels. But let's go ahead and create a panel. So to create a panel, we'll head over to workflows and panels. And here there's a drop down. You can either create a panel or create a custom workflow. So let's go ahead and create a panel. And by hitting create, then step 1 is the panel name. And then project. When I create a panel, I always have to assign it to a project. As a reminder, projects are children of groups. And so only people that are in that group will have access to this panel. So I'm going to assign this to my group. And then I can either select a panel to use as a base, another panel, either one of my custom ones or one of the OMIPs or one of the BD panels. I could select to use as a base and make modifications, or if I don't select any of these, it'll just mean I'm selecting one from scratch. So let's do that as a first go. So I've named the panel, and on this step, I select what cytometer I am planning to run this panel on. So when I click here, I see the list of cytometers I've uploaded to my organization. Again, it could be one I created or just one that I've linked to from a core lab user. And I'll just, select this A5SC. And if I expand it, I can double check that, yes, this is the configuration and cytometer I plan to run this panel on. So I'll complete step 1, which is selecting the cytometer. Step 2 is selecting the target. So what are the markers that I want to create for my panel? So here I can either choose to add markers one at a time, right? CD3, CD14, and so on. And I can assign antigen density one at a time. So if this is a monocyte panel, I'll say it's CD3 negative, CD3 either generic positive, or I can say low, medium, or high. This will help me in the next step to choose the brightness of the fluorochromes based on antigen density. So that's option 1, to either go one at a time, or like I said, in the population section, you can predefine these for your lab. So in my case, I'm going to go here, EJ monocytes custom, and press add. So now it will add multiple markers all at once with the information I've put into the population section, right? So it already came pre-populated, negative, positive for those markers. I'm going to go ahead and delete this default population. So I've imported EJ monocytes. If I wanted to create a particularly large panel, I can put additional cell types. let's see, cell populations. Let's do T cells. CD8 naive T cells, for example. So now it'll create not only additional markers with a defined antigen density, but it'll also add unknowns if those were not predefined in the population section. So now I have two populations. I have EJ monocytes and CD8 naive T cells, and I can add multiple additional populations that way. That'll come in handy in the next step. But for now, I'll just delete it. And I'll keep this EJ monocyte single population. I can include additional viability dye. If I want to create a dump channel where I put multiple markers on the same fluorochrome to eliminate them for negative selection, I can choose to do that. And I often do that for B cells. So in this case, I'm going to select CD19 and CD20 to be put together under exclusion as a dump channel. Other things I can do on this site, this lets me know I'm using 9 fluorochromes out of the 48 available ones based on my cytometer choice. And I can also click on this button if I want to restrict which reagents are available to me during panel creation. So if I only need a certain category, I can uncheck these, or if I want to maximize fluorochrome options, I'll leave those all checked. Other things I can do, if I have a favorite clone that I want to restrict choices on, I can choose to do that, or if I just leave it on generic, it'll show me all the fluorochrome options available. Any questions so far for this step 2? If not, I'll continue to step 3. So step 3 is where we will perform pairing of the markers from step 2, which are listed here, and the fluorochrome list. So if I click on this first antigen, I can click on the various fluorochrome options, and on the spectrum viewer, it'll show me either the spectral signature, if I've selected a spectral instrument, in which case I have here, or if I don't want to view the spectral signature, I can always untoggle this and it'll show me the traditional view. So here I can go through and see the different markers, the different fluorochromes that are available to me. And based on the configuration of the cytometer, I can see if this is a good marker or not for my specific cytometer. So in this case, what else can I do? I can do in the antigen name, I can sort this list in different ways. I can sort it by antigen name, and then sort high to low, low to high, or I can sort by antigen density as well. So for those that are defined here, I'm saying CD14 is very bright, or so it has a high antigen density. And as we know, we want to have high antigen density with low brightness fluorochromes and high brightness fluorochromes with low antigen density. So I can actually sort the lists using these criteria. So brightness, right now you can see the brightness here, 1, 2, 3, 5 being the highest, 1 being the lowest, or I can sort them in reverse. So for a high antigen density, I'll want to click something that's relatively dim. Let's go with this one just for fun. I can also choose to sort them by fluorochrome availability. So this shows me high to low that CD3 comes in the most colors, or if I flip it, the viability dye comes in the fewest available fluorochrome. So you might want to choose to build your panel in that way as well. So CD197 in this case, I might want to build my panel around this one because it doesn't come in as many colors as the other ones, although we are seeing that there are quite a few colors here. Again, this list of fluorochromes, I want to remind you, gets populated in 2 ways. One from the BD catalog, so you can see that we have quite the selection. But if you wanted to also add additional ones from the custom reagent here for CD3, I added some Nova Fluors that BD doesn't offer, or some Sparks. Those were all created in the custom reagent and they show up for me during panel creation. If I had any reagents in my inventory, I can't remember if I did this or not. I think I did for my FOXP3. I didn't think to add it here. But if I had reagents in my inventory for any of these, I would have a little check mark here letting me know you already have this antibody. So that could be also useful information if I'm hesitating between two fluorochromes that are very similar to each other and I already have one in the fridge. Well, then it would make sense for me to select that one. So if you have the inventory feature, that comes along with that. I'm trying to think what else here. So this, you can sort by name, alphabetical. You can also, you don't have to scroll the whole list. If you know you're looking for a specific fluorochrome, you could just type it in. So of course, this one doesn't come here, but let's say here, if I'm looking for APC, here it is, CD45RA. So right now I'm just making random assignments for the sake of time. But if you were truly creating a panel from scratch, you would spend a bit more time evaluating the different fluorochrome and filter combinations. So here, as I'm making pairings, the fluorochromes become static, right? So as I'm clicking through here, they disappear if I don't select them. But as I click pairing, then they become permanent. And as I add more fluorochromes to the list, I get to see the interactions of these different fluorochromes, letting me know if I'm going to have, you know, spillover spreading based on spillover values. And as well, if I've made pairings up here, some of these become hashed, telling me, okay, this reagent CD8 is available in APC, but I've already paired something else to APC, so it's unavailable. So it's hashed here. Similarly, this red box, if I hover over it, it says APC size 7 uses the same peak filter as APC H7, and they're not recommended to be used together. It doesn't prevent me, so I can still click it and still pair it if I so choose, but it's telling me that these two have high similarity and use the same peak detector, and that I probably don't want to make this choice. So if I don't want to see these red box fluorochromes as options, I can say hide occupied filter floors. If I don't want to see the paired ones, I can say hide the pairs. So what's remaining is only fluorochromes that are better choices for me. And then same with inventory features. If I toggle this, it will only show me what I've already have in the lab as options for me. If I come back to spectral flow here for spectral instrument, I get to see the spectral profiles. And then I can also toggle to look at the similarity matrix. So if I'm creating a spectral instrument and I'm clicking on these different fluorochromes, it will update this table and show me the similarity index for these different combinations. And as a reminder, as you get closer to to 1, it means the fluorochromes are similar to each other and aren't great to be used together in the panel. Oops. All right. And then the populations table here shows the different populations that I've chosen to include. So EJ monocytes, if I had NK cells, T cells, DC cells, I can have multiple populations. And the advantage of putting them all listed here individually is, let me just finish this panel real quick by pairing these. So just with random assignments. Once everything is paired, I can choose to remove from view one or more fluorochromes if I want to inspect the profile of what's left. But here in the population table, what I can choose to do is view this population, view the fluorochrome profile of this view the profile of this population. So by clicking it, what it will do is it will hide, in this case, CD3, CD8, viability and exclusion markers. So anything that's negative for this population, it will remove them from view, from the spectrum viewer. And all that's left are the fluorochromes that are being expressed by this population. So now I can more closely inspect this population to see, all right, do these fluorochromes, the remaining fluorochromes, do they interact with each other? And it'll give me an idea of spillover spreading, right? And I can also change the view here and go through laser by laser. So here I'm seeing the spillover spreading with the hashed lines, or not spillover spreading, excuse me, cross laser excitation. And the violet, which ones are going to be excited there, which ones are going to be excited here, same here and here. So this is just showing me only the fluorochromes that are being positively expressed in this population. So if you had multiple, you can go through one at a time and inspect your fluorochrome choices more closely. Any questions about this view? Step 3 of panel creation. So this is where we expect most people to spend time carefully evaluating fluorochrome combinations here. So once I'm done and I've paired all the markers from this list, I hit complete step, and this brings me to step 4. Step 4 shows me the reagent options for the fluorochrome decisions I've made in the previous step. So CD3, Alexa Fluor 700 was a combination that I chose in the previous step. And if I click on this down arrow, it shows me what reagents are available from the BD catalog for this combination. So you can have different clones, different test sizes, and you just go through and you select which ones you want. Some are have more choices than others, right? So if you don't go for a smaller test size, you can. The pricing we put is the list price. Of course, your pricing may be different based on your institutional discounts. So I'll show you how to see those later on. So once I've made the decisions of which clone and test sizes I want to move forward with, I hit complete step on step 4, which brings me to step 5. At this point, I've finalized the panel. I've decided which clones I'm moving forward with. Here I am finalizing the shopping list. So if I have items that are already in my inventory, they don't need to purchase, I can delete them from here. And so they're going to be removed items. And then the only thing that I'm leaving is items I need to purchase. So then here I see the estimated total of the list price, and I can finalize this workflow. And then it will take the items from my shopping list. And if I click add to BD cart, it'll bring me over to the BD website, add those items to the BD cart. And if I log in at this point, then the list price will be converted to my actual price based on my institutional discounts. If you don't have access to purchasing through the BD website, not a problem. You can also download this list. So either the entire panel or just the shopping list, either as a PDF or a CSV, and you can download it and share it with anyone who has purchasing privileges in your lab. Similarly, you can email these as well. Alternatively, you can also request a quote. So by pressing this button, it'll automatically send a list of these reagents to your local rep, and they will contact you with a quote for these reagents. So once you are done with panel creation, this panel will now live in the panels resource under custom panels. So here's the panel that we just created. If I click on it, here it is populated with the different fluorochromes on the different laser lines that I've chosen. And then here's the cytometer used to create this panel. From this point, once I receive the order of my reagents, if I have access to the inventory feature, I can just come in here and click add to inventory. Once I've titrated these, I can add the titer information in the inventory, and then I'll be ready to run this panel. And you can, easily link your panel examples to this panel using the linking feature for easy access if you need examples of titrations or full panel information. Once you run out of these and you need to reorder, you can easily do it from here, order reagents. It'll bring you to the similar steps that we just went through, right? So you can delete the ones you don't want and then get to that final list quite easily without having to copy paste a bunch of information. What else? You can download PDF, download CSV. You can delete this panel if you no longer use it. Or alternatively, if this was a first iteration and it looked okay in a few markers, but a couple of them were causing issues, you can use this panel as base. So if I click this, it will now trigger the same workflow we just went through, but using this one as a base. And there are a couple of decisions I need to make up front. So I'll just rename this, example 2. Again, I have to assign it to a group and a project. And then I have to decide, do I want to keep these clones because they're important, or I don't really care about the clones. I want to see what other options they have. So I can either remove the clones or keep the clone information. And same with the pairings. Do I want to keep the existing pairings, or do I want to remove them because all I care about is the antigen list, right? So you can choose to keep or delete certain features, or you can choose to remove a few of them before proceeding to the next step. So if I just leave everything as is, I can always reset. If I made some changes and change my mind, I can reset it, come back to this setting, and then continue. So now it just pre-fills. It brings me to step 2. Step 1 is reusing that same cytometer. I come to this step. It already has the clone information pre-filled. It has the antigen list. I can add to these antigen lists. I can remove them. And then step 3, it'll just show them already paired on this step. If I want to unpair fluorochromes that didn't work out well for me and see what other options they have, I can do so. And you would just continue, and then you would end up with a second panel. Another thing I did not mention is if ever your cytometer goes down and you need to see if your panel, is compatible with a different cytometer, you can use this feature as well. So here I could use this panel as base. And then it shows that this is the cytometer I created on, but if for whatever reason it's unavailable and I need to see, okay, right now I only have an A3 that's available to me instead of an A5. Can I use this? Yes or no? And if I continue, if it's not compatible, I'll get a little warning. In this case, I did not get a warning, so it shows that it's compatible. I would just proceed to this step. Now it'll show me the new cytometer configuration. I get to decide, I get to see the different fluorochromes based on these new filters and see if this is a good compatible fit or not. Okay, so that was panel creation. And I see that we're running out of time. It's going by very quickly. Very briefly, I'm just gonna talk about the File Explorer. This is where I can upload and save data. So all these different folders are the different projects of this group. I can click on a different folder and see the project files within it. And I can see that there are different FCS files, FlowJo workspaces, JPEGs, PDFs, any data that I have related to this project, I can upload here directly in BDRC. Nice part is some of these I can just view directly in BDRC. So view this PDF. Other things I can do for individual files, I can delete them, download them, share them outside of my organization. This is a premium feature. Or I can link certain files with other resources. So I can link this to a reagent, I can link it to a panel just for easy access to stay organized. For FlowJo workspaces, I can also choose to lock them. So if I'm working on them and I want to ensure no one else is looking into this workspace at the same time as me, I can choose to do that. I can email it, things of that nature. So I will just kick off FlowJo to show you how this starts. I have a FlowJo portal, but you don't have to. If you have a dongle license or serial number license, you can still connect to BDRC. You'll just have to enter your BDRC credentials separately. For me, since I have a portal account, I just enter that information at this point. Trying to remember what my login is. Okay. So once I'm connected to FlowJo, I can add samples from BD Research Cloud. So I don't even have to have BD Research Cloud open. This could be all gone. Whoops. And then, so after doing add samples from BD Research Cloud, I see this little modal pop up. And so from here, I can select which organization I want to pull data from. So here I'll just switch to my lab organization. I can click to titrations. And then I'll be able to view all the FCS files that I've uploaded directly to BDRC. So in this case, I'll just select these 3 files. I'll say add. And what BDRC will do is download these in the background. Whoops, did I not click? There we go. It'll download those files in the background so that the files are actually local. So right now, FlowJo is not connecting to BDRC and performing analysis through a server. It is performing analysis through files that are local. And in case you're not aware, FlowJo really does have optimal performance when the files are locally stored. If your files are up in the server, then that lag in connectivity can really affect performance and may even cause some crashing issues. So we always recommend that you have your files locally stored on your computer for optimal FlowJo performance. So BDRC has that built in, that even if you have your files available to you from anywhere up in the cloud, you can connect FlowJo to them, and then they are managed in the background without you having to copy and paste and repoint, to the new location. Similarly, if you've already performed analysis and you're already at this point, the files are local and you have not uploaded them yet to BDRC, you can do that as well from FlowJo. So in this case, I would just do File, Save As Workspace to BD Research Cloud. So just click this. Again, the same little window will pop up to ask me, where do you want to save it within BDRC? I would then select the organization, the project, and the right location. Here we go. So in this case, if I want to save this workspace to my demo project, and then I can select a subfolder, I can create a new folder, I can save it within a workflow, hit save, and then it gets saved automatically to BDRC. If the FCS files are already located in BDRC, it does not create a copy. It already knows where to find them. But if they have not been saved yet, saving the workspace will also save the FCS file, so you don't have to worry about that. You don't have to upload the FCS file separately. It'll just package the whole thing and save it for you. So that was the connectivity piece of FlowJo to your BD Research Cloud. The only other thing that we have not covered was creating a FACSDiva experiment. So that would be a resources, go to experiments. You can create a new experiment. So you have to name it, assign it to a project to stay organized, select a cytometer you want to create it with. So let's go again with my A5SE, select the Diva version. And mine will have a plate loader, so I'll just select to start that way. I would select a size of the plate. So in this case, it's a 96 well U bottom. I highlight which wells I want to add samples to. I like to use the autofill selected wells. I'm going to select a pattern. So each specimen is going to go down for 3 wells. So autofill, so now my specimens are properly labeled. I would continue to the next step. This is where it's really nice. You can select all your samples and pull in a panel. So let's go ahead and pull in a panel that I made back in March. I'll say apply panel. And what it'll do is it will automatically label all your parameter channels for you so that there's no typos, everything's assigned properly to all your samples. You can continue from this step on. We always recommend adding keywords to samples. So keywords can represent stimulation conditions or any kind of additional information that's relevant to your sample. Maybe it's the age of the different samples that you want to then chart later on. The reason why we encourage users to add this information prior to acquisition is that once you record your data, that information will be embedded and it will stay within the FCS file. So that's how you avoid ending up with samples or experiments that have FCS files that you have no idea what they are. You know, the channels aren't labeled, you don't know what the samples are, what the different conditions are. So just doing it ahead of time quite easily. So this has a spreadsheet-like interface that you can copy and paste with one click large amounts of information. So again, we recommend doing this prior to acquisition. It'll get embedded into the FCS. Next step, if we go into plots, this is an easy way to add many plots. You can do it N by N, so it'll show all the colors versus all the colors. You can choose to include all parameters of your cytometer or just the ones that are labeled, depending if you're doing spectral cytometry or not. Or you can also choose to create a gaining hierarchy. So in this case, forward and side scatter, I'll add a polygon to the middle of it to create this P1. I can rename the P1 to call it lymphocytes. Then I can add an additional plot, say another dot plot, and then I can change the parameters to say CD45 versus, whoops, I forgot to hit enter. CD45 versus CD8 on my lymphocyte population, and you can create your hierarchy ahead of time. You can do polygon, rectangle, intervals, and decide where to place it. In BDRC, these are static, so you can't draw them or move them around. But once they're imported into Diva, you can definitely move them around as needed, and also stretch out the vertices to make custom shapes at that point. Lastly, you have cytometer settings. So you can change your cytometer settings ahead of time if you have set values you want to get closer to before running your CS&Ts. If you are not running spectral, you can choose to remove the unused parameters to only collect parameters that are in your panel. And then once you're done, you could just save this, and it creates an XML that you could then import directly into Diva. So this is a nice time saver that you don't have to, especially if you're paying per time on the cytometer, you don't have to spend time labeling your samples. You can do that ahead of time and save how much time you spend on the cytometer. And this is also good for core lab managers to encourage use, because then it'll give you more users per day for your cytometer if they're not just sitting there labeling their samples, if they're actually acquiring samples. So that was the overview of BDRC. I hope you found it informative. This webinar was recorded, so there will be it will be available on the FlowJo site. I'll show you where. So if you go to flowjo.com under Learn Webinars, this was today's webinar, but if you scroll down, we also have other upcoming interesting webinars. Feel free to sign up for those. But then here, this explore previously recorded webinars, you can click on here. So this webinar will show up here at this point. Other resources available to you is FlowJo University. If you click Learn FlowJo University, then you have our different products. If you click on a BD Research Cloud, then you have access to short bite-sized videos. So just 1 or 2 minute longs of different sections of BDRC. If you just want to quickly be reminded on how to perform certain tasks, that's available there as well. And then lastly, back into BDRC. If you have a question here, there's a little tech support icon that you can click on. So BD Research Cloud Technical Support. So if you need help, it shows you where those videos are, the FlowJo University. We have frequently asked questions. It reminds you that you can click tutorials at any time to get more information. I'll show that to you in just a bit. If something's not working or you need additional support, feel free to reach out to flowjo.com, or if you have future requests, feel free to email me saying, oh, I would love it if BDRC would do this. As a reminder, BDRC is in active development, which means we're still adding new features. We have a release coming up in November, which is going to be really exciting. So I would strongly encourage you to check back in to see what we have. We're going to be able to scan some reagents using your phone. We have some staining sheets on how you can pull in titration information from your reagents to come up with staining protocols with titers. So yeah, make sure you check back in for that. So yeah, if you want to see additional features that aren't there yet, just pop it into the comment box and I'll be getting those. And then here, this tutorial is where you have additional information. The content of the tutorial changes depending which page you're on. So the first page when you first show up is this one. It's the workflows page. So this one has a little overview video, 2 minutes long, of BDRC, the main features. If you scroll down, it also informs you of different information that might be relevant for the page that you're on. Thanks again for attending today. I'm happy to take any questions. If not, I hope you give BDRC a try and let me know what you think. Thank you so much.